welcome to the SquaMates podcast. This is a very serious podcast about herpetology, and I am one of your hosts. My name is Dr. Mark D. Schertz. I'm the curator of herpetology at the Natural History Museum of Denmark, and I'm joined today by three fine co-hosts of mine, Ethan Kosak.
Hi, I'm Ethan. I am an artist, an illustrator, and reptile enthusiast.
Gabriel Lughetto.
Hi, I'm Gabriel Lughetto, and I'm a paleo artist and scientific illustrator and concept artist, and I used to work in herpetology, but not anymore.
And Hiral Naik.
Hi, I'm Hiral Naik, snake ecologist and conservationist from South Africa.
Wow, we are a greased machine. It only took us, 20... We've actually recorded 32 episodes, I think, by now, but only, I think, 28 of them have ever aired.
There's a couple of lost episodes.
There are a few lost in the vault and hiding around on various external hard drives of mine because, of course, a lost episode still takes up a lot of space.
And a couple of failed attempts, too.
Several failed attempts, honestly. You'd think that we'd have mastered it by now, and yet here we all are. But it is, as ever, a pleasure to talk with you guys about the wonderful things of herpetology. And so, maybe we can start off with what's going on with you guys since the last time we recorded, with I think Ethan, you just established was July. Is that right?
Yeah, it was the end of July. Yeah.
Gosh, it's been a time. It's been a time.
Yeah.
Been a while.
Yeah. Well, I crowdfunded a book, and it is being made, and I'm going to be doing that forever.
Yeah. Yeah. I mean, it's a massive commitment, but your crowdfunding was unfortunately successful, right? I think.
Oh, just horribly successful.
Yeah.
And now I have to do that. So that's what I've been doing. And yeah.
That's often the tone that people have with grants as well. They're like, "Oh, well, I got the grant, and now I actually have to do the thing."
There's an early Simpsons episode where Homer's in space, and he crashes into an ant farm that they have, and it's translating what the ants are saying. And the line is, "Freedom, horrible, horrible freedom." And that's what I think of when,
Yeah, that's actually really relatable. So I guess I'll go next because this is directly alongside what's happening with me, which is that I got an ERC starting grant, which for those of you who don't know, an ERC starting grant is a research grant by the European Research Council for 1.5 million euros, which is a lot of money. And they are very, very, very competitive and illustrious and whatever, I suppose. I'm kind of sick of hearing it by now. Everybody at the Institute here has been incredibly kind and very supportive and very congratulatory and everything. They're like, "Oh, we should mention it at this meeting, at that meeting, at this meeting." And so by now I've heard it 10 times. And if they keep talking about it when I'm not there, it's fine. But while I'm there, I'm just like, oh God, because because the grant will start on the 1st of January. And every time they mention it, I'm just like, oh God, I have to do it. I've got to actually do the thing. Which, of course, is a huge pleasure. So in brief, the project is called Gymmie. It's on the genomics of miniaturization of vertebrates. And essentially what the conceit of the project is that by sequencing reference quality, so very, very high quality genomes of a whole bunch of independently miniaturized vertebrates and closely related non-miniaturized vertebrates, we'll be able to establish through that comparative framework, those pairs, establish what are the general rules, what's happening every time something gets miniaturized, and then perhaps even going so far as to saying, okay, what is promoting miniaturization consistently and what's stopping things from miniaturizing? So it's a very complex set of things that we'll be able to do with all of these genomic resources. And then on top of that, they serve kind of the baseline data for everything else that will happen in my lab from this point onwards. So it is a total change of direction for my lab. I mean, longtime listeners of the podcast know that I've been very interested in genomics for a very long time. But an opportunity for me to go whole hog and and go, across the board. I'm not just doing herps. I decided to go, to make it broadly appealing. I wanted to go at a larger scale, but of course get herps in there. And so it seemed logical to go and take all vertebrates and kind of hit them all with one fell swoop. So it goes everything from sharks through to frogs, if you want to twist the tree that way. Lots of amphibians in there because amphibians have done it so often, but it's a huge data set. I've got at least 40 pairs of miniaturized and non-miniaturized, 20 pairs, so 40 lineages of miniaturized and non-miniaturized species in there. So, and we keep adding more. So it just applied for an additional tiny bit of money to sequence the world's smallest tortoise, which is very, very small indeed. Padloper from South Africa. And we'll see also some people just wrote to me about pygmy seahorses, which are also super. So
I've kept pygmy seahorses before. They are
cool.
interesting. Yeah.
Very cool. Yeah, so that has been crazy. And actually, in addition to that, we just got another pot of money. So that will give me a PhD student, a postdoc, and a research assistant over the course of the next 5 years. And of course, 1.5 million euros that I can burn on genomes. And much of that is really going into just the sequencing pot. But then at the same time, we applied for a different proposal where I have a postdoc built in. So, and this one was also successful. And the project there is essentially to develop a high throughput micro CT machine. So we are going to I will eventually get a postdoc to deal with several hundred micro CT scans of SquaMates over a 1-year period, roughly, because that's how much money was available. So it's going to be an intense thing, and it'll come only in a couple of years once the machine has been built. But that's 2 fairly large grants and projects that have gone through in quick succession, which is nice after a very long dry period between grant success. So, yeah, that's the biggest updates for me. We'll have one more update from me after the break in a bit. But how about you guys? Hiral, how's everything?
Yeah, I've also been pretty busy. Mark and I met at the World Congress, but we'll talk about that a little bit just now. The biggest update on my side, though, is that I am basically a week or 2 away from submitting my PhD for examination. So,
Congratulations.
Which means by the time this airs, it might have already happened. Oh my goodness.
Yes.
Wow.
Yeah.
Oh my goodness. So what's the protocol in South Africa for PhD submission, defense, etc.? How does that work?
So we don't do an oral defense. We have to get one internal examiner, which is usually within South Africa, and then 2 external examiners from some other part of the world. And they get 6 weeks to read through the PhD thesis and then decide whether or not that's
sufficient.
Yeah, ready. Yeah.
Okay, and if they come back with revisions, do you have to make those revisions before you get the degree?
Yes.
Okay. Yeah. Okay. So it's quite a lot like the old Australian system, if I understand that correctly, right? So only recently Australia introduced oral defenses. I believe they had until recently. It was all just as there is apparently in South Africa as well. Yeah, I could be mistaken here, but I've heard that recently. One of my someone I was speaking to recently, can't remember who it was, said that they were one of the first people to defend as an oral defense at their university. It was a friend of mine, Raul, who was here. He works on corals.
But you have to do it upside down.
Yeah. Of course. Yeah, wicked. That must be such
because of the Coriolis effect.
Yes, it's true. You have to spiral. I guess everybody goes through a spiral during their PhD, but in the South, you go the other way.
Yeah.
So it must be a huge relief. What's happening after that for you? Are you taking a break? Are you hitting the...
Yeah, it's a little complicated. I've got grant, I guess, commitments to live up to.
Right.
and I'm working on another paper and all sorts of other things. I have a conference, the Herpetological Association of Africa conference is happening at the end of November. So there's a couple of things. I don't quite know. I think I can squeeze in some downtime before that.
Yeah.
And then I'll make the next announcement at the next episode.
All right. Oh, that's exciting. That will also have to wait for a little bit, because one thing I forgot to mention is that I've also booked my flights and everything. I'm going to Madagascar again from the 3rd of November, I think, until the 8th of December. So 5-week trip that's coming up soon. But yes, that's... Yes, when I finished my PhD, I found it incredibly valuable after submission, but before defense, to go and take some time. And actually, I went to Madagascar. It was the last time I went in 2019. So it's been ages and ages since I've been back to Madagascar. And in the intervening time, I've been to like New Zealand and Borneo, but not been back to Madagascar. But in any case, it was super, super good for my like mental health and feeling of wrapping it up, to take a little bit of a break and, do something for me that was not, of course, it was also an expedition. We did a bit of collecting, but it was for fun, so that was great. Yeah. And Gabriel, how about you?
Yeah, I've been super busy. I'm actually kind of overwhelmed with how busy I am. Well, first of all, we're recording this in October, and this is going to come out in October too. So I'm doing my series of October ink and marker drawings every year, which is a nightmare because I don't have time, and I have to squeeze time in the morning. Like today, I only had time to do some, and I have to finish that after we finish recording. So I'm kind of like...
I'm doing one too this year. But I'm doing Skinktober again.
Skinktober, yes. Skinktober is great.
Cool.
It's hard to find the time.
It is. It's a stupid thing that I'm doing,
Yeah, same with me, but everybody seems to like it, so I have to do it.
Where can one go to see the Skinktober and your Inktober things, Gabriel and Ethan? Are they
doing Blue Sky. I'm doing
Blue Sky?
Blue Sky. Yeah.
Yeah, I'm all in all my social media.
All
you can see it.
Okay.
In all of them. And then like every year they will be on sale later, probably early next year.
Mm-hmm. Handles are at the end of the episode for everybody who wants to go and find that content.
I have a couple of yours from other years that I've
you
purchased.
do? It's true.
Yeah.
Every time I've wanted to buy an Anuragnathus, someone else has beaten me to it. So
yeah, I mean, what happens usually is that what I tell people is that if you like one of the pieces when I put them out in October, just reserve them, and I will reserve them for you. And then when I do all the shipping and selling all that in early next year, then I'll have your name written down for one of those. And because I'm very happy to say that they sell out very quickly.
Yeah.
And then I've been working on a lot of things, a lot of commissions that are all under embargo, but some really cool stuff. One of the things that came out recently was an illustration of a glyptodon that I did for the University of Wisconsin Geology Museum. I did that 5 years ago, and the exhibit just came out now. So it's been under embargo for 5 years. That shows you what what the embargo times is for some of these things. I did an illustration for that, and also I did some drawings of the osteoderms of the tail of the glyptodon, showing how they correlate with bone structures of the caudal vertebrae and all that. That's what I've been doing. I'm working on a lot of stuff right now that I cannot, talk about yet. Also, the book the illustrations that I did for the book of, I think I mentioned this last time, but I did several illustrations for the new book on dinosaur behavior by Dr. David Hone, published by Princeton Press, that will come out late in November 5th, but you can pre-order now. So if you're interested and you want to see a bunch of my illustrations and drawings, and Dave is a super well-known paleontologist, and
also has a
wonderful
podcast, book. Terrible Lizards.
Yes,
together with Izzy
Lawrence,
who's also hilarious. It's very enjoyable.
So yeah, that's what I've been doing.
Awesome. Yeah, I mean, it's always like, I don't know how it is for listeners, but every time I listen to what you guys have been up to, I'm just like, oh my god, everybody is doing so much all the time.
Listen, you have to pay the bills. You have to do it.
Oh my god, yeah, absolutely.
Yeah.
The bills are not getting any cheaper. So
that is so true. And I've been really
or paid, really. I mean, I don't know what I do.
Yeah, I've been really having a rough time with email recently because I was out for a week. I got shingles. And it sucked. And then I was basically for over a week, I didn't answer any emails. And then I came back and I'm still like, apologies to anybody who's waiting on emails from me from July. I only got the shingles in September, but like I'm going to use that as an excuse for everyone now. I'm so far behind.
I know what skink I'm going to draw today, Mark.
Oh, shingleback? Excellent skink. Very good skink. Are they in Taliqua? No.
Yes,
I think
they
so,
are.
Yeah,
okay. Taliqua are bonkers. Yeah. What a genus.
What a genus.
This has been. I mean, this is one of the fun things that we have going on right now at the museum. Is we have Haley Crowell, who's actually a rattlesnake visual ecologist, is here doing an internship with me at the museum, and at the moment she is in the process of reorganizing a huge amount of material in the collection, which is super, super cool. So she redid all the geckos recently, and now it's what started off as maybe we should fix some of the colubrids because the genera and the families are a bit of a mess, is now escalated to she's redoing all snakes. And it's like we have some 40,000 snake specimens. So it is no small task to reorganize all of the snakes, but it's brilliant. It's going really, really well so far. So I'm very happy to have.
Isn't that how all projects start? Because that's how they are for me. I start with a small thing, and then by the end, I've grown into a gargantuan thing that makes me want to kill myself.
Your book, and that of Darren Naish is under a kind of similar timeline, I suppose.
Although I must say, I've let me knock on wood, but I've done some pretty good, solid progress finally.
Glad to hear that. Yeah. Very glad to hear that. Anyway, I think one of the things we wanted to talk about next was a bit about the World Congress, because obviously, kind of unannounced into our feed, at the same time released into the Herp Highlights feed, people will have heard Hiral and me catching up with the chaps from Herp Highlights, which was really fun. We had a great time recording in an empty lecture theater that then people kept like walking through, so occasionally someone walked through the background of the video. But it was... yeah, Hiral, maybe you should summarize it. I think I'm lost for words.
Oh man, it's hard to believe it was over a month and a bit ago.
Yeah, crazy.
But it was crazy. There were tons of people. I don't think I've ever seen so many herpetologists in one space ever. And a lot of well-known people, a lot of in different, their different realms. And it was Borneo. So it was really as expected, going to be one of those big events. And it was quite exciting. I mean, it was really great to see so many people that I'd met at other conferences. So this was my third time attending the World Congress, which was also really cool. It was great. So when I first attended the one in China in 2016, I was one of the students among some of the other students, but now it's so great to see so many people that have, achieved so much. Feels like we're actually growing up. We're doing some herp stuff.
Yeah. I think that's awesome as well. Like seeing the young people in 1 year and then 4 years later, it's a different stage. And because it's every 4 years, you're seeing them like oh, it's a bachelor's student. Oh, they're in their second year of their PhD. There's a totally different perspective to where they were last time you saw them, or
and also a lot more tired.
Yeah, of course. I mean, everybody's also more tired. The last one was pre-COVID. It was January 2020. So we got back, and I was reading in the plane as we got back, like the first cases of this weird sickness that had gotten to Germany. It was like one town away from where I was living, and it's like that was it. Like it was just before the pandemic. So the last one was kind of this idyllic. Nobody gives a damn about anything. Everybody's intermixing and everything. And
a simpler time.
This one, you could tell, things have changed a lot. The way the everything works now has changed a lot. The masks, there weren't very many people wearing masks initially. That changed very fast over the course of the week, to the point where at the end of the week, like on the first days, I had arrived and gotten a tiny bit sick. Specifically, I developed like sores on my face because of the Malarone that I was taking to prevent malaria. So I was like covering it with a mask, hiding. And then I was mostly wearing that when I was together with lots of people. I would then take it off in between, but like a bit dynamic, which is obviously not the best thing that you should do, but it was better than nothing. And by the end of the week, I would say a third or more of people had a mask on at least at some point of the day. So like it really, because there was a lot of coughing and stuff. And these events every single one of them is a super spreader now. So yeah, that's just how it goes. Unfortunately,
just getting on an airplane, you're asking for trouble.
Absolutely.
Yeah.
I count myself very, very lucky for not having gotten sick, like not getting COVID or flu or anything on this trip, because every trip I've taken recently.
We did a trip over the summer, and it was like a 3-hour flight. It wasn't like a big flight, but even that, like, I'm actually shocked that none of us got sick, because you're just getting into a tin can full of disease,
That's it, right? I mean, yeah.
There's no way I get into a plane without a mask, and I don't get into an Uber without a mask. So.
Yeah.
And I haven't gotten COVID, so let me not go ever.
Wow. I think I've had it 5 times now, but that's
what?
Yeah, because I ride a full train every day, and I've got a kid in a daycare who brings home a fine selection of all the illnesses in the community.
We've only had it once, but I will say, going back, our kid was in daycare before COVID, but even then, like, that was probably the sickest we've ever been continuously.
Yeah, rubbish. Yeah. Well, not to get too much into the COVID weeds.
If I could be wearing a hazmat suit every day, I would.
Yep. Don't go near children. That's all I'll tell you.
That's not a. Yeah. I know that they're like little petri dishes. I don't know.
Mm-hmm.
All right, let's bring it back on the rails. No, I think I thought, and I love the World Congress. I think it's an amazing meeting. One of the things that we didn't know at the recording that Hiral and I did, it hadn't yet been announced where the next meeting is going to be. It is going to be in Spain. And that is very exciting, of course, because it means that it will be easier to get to for many people. Although, unfortunately, the location that's been chosen requires a flight from practically everywhere, including Europe. So,
where is it?
It's in Gijón, close to Portugal, northwestern Spain.
That's going to be like, it's in the Strait of Gibraltar.
Oh, no. No, I mean, I think we're basically at a point where this meeting essentially needs to be held in a large city. And one that has a major international airport, because doing inland flights is just bonkers. But anyway, this one will indeed, although I think there was some disappointment among many people that it couldn't be in Nairobi, or that they chose not to put it in Nairobi. I think for this time, it certainly will be an exciting event to have in Spain, and I'm happy that it will be in Europe again for many of us. And I think also beyond time that we have another meeting in the US. So that I hope will be in a few years, or in the the coming ones, just because it's been so often now East, and the Americans, who make up such an incredibly large portion of the attendees, are then having to fly like crazy no matter where it is. So yeah, to some extent we do have to be thinking about the footprint of these things, and it's good to organize it in a way that kind of works. But it's great that we have this this rotating schedule of continents and countries and everything. So that seems to be working really nicely. And yeah, I think on the whole, the meeting was amazing. And only, I mean, I always find the pieces around the meeting much more exciting than the meeting itself, because I'm just not so keen on talks, especially when you have to ram yourself into a room that's too small with a very full crowd. So it's just amazing to get to hang out with such wonderful people. To see her all in person again was so nice, and to hang out with so many other people that I'm so very, very fond of. So we have to see. Hopefully, when it does come up to the US, and if we are still in business, then we'll be able to get you guys there. Perhaps even in 4 years, we can somehow find some funds for podcasting, social media outreach type of stuff, and get us all to the same place. That would be so cool.
It'd be like a harmonic convergence of
it would be. It would be. But I think there's, these kinds of meetings, if you go and look at like the paleontology meetings as well, they've done a really good job of integrating paleo podcast people and doing those things. And I think the guys from Herp Highlights, Tom and Ben, they did such a good job of representing kind of herp outreach stuff. And it would have been nice if we had also been more proactive on that. But of course, I think Hiral and I were both coming in there with our like research hats on. So, but yeah, thoughts for the future. In any case, from here, I think unless there's anything else we want to say, we are going to take a short break, and we'll be right back after this short musical interlude to talk about some very exciting new frogs and some scuba diving lizards and some Gymmie. So,
new frogs just dropped.
New frogs just dropped, and they're pretty cool. Okay, we're back. And I'm going to start off this part of the whole thing with some breaking news, which is a new paper that is published today in the journal Vertebrate Zoology. Today, assuming today is the 14th of October, and assuming that they do actually accept the 14th of October as the publication date. But assuming those things, today, Vertebrate Zoology, 14 October, new paper by Miguel Vences and colleagues, including myself.
SquaMates exclusive.
Except not exclusive because it's going to be there's a press release anyway. But I'm here exclusively talking about it to your fine ears. First author Miguel Vences, myself as the last author. The paper is titled "Communicator Whistles: A Trek Through the Taxonomy of the Buphus marigesensis Complex Reveals 7 New Morphologically Cryptic Tree Frogs from Madagascar." There are some hints in there, dear listener, as to what we have done, because we have just described 7 new, beautiful tree frog species from across the rainforests of Madagascar, and we have named them, see if you can spot the pattern, Buphus kirki, Buphus picardi, Buphus archery, Buphus janeway, Buphus pikey, Buphus ciscoe, and Buphus burnhame. We have named 7 new species after 7 Star Trek captains, because we are huge nerds.
Congratulations, first of all.
Thank you so much. This is something that Miguel, first author, and I have been talking about since I was a master's student, because, at one point I stayed at his house in his flat while I was doing work in Braunschweig, and in his living room he has a life-size cutout of Captain Kirk. And that is when I discovered that I was in the company of another Trekkie. And so Miguel and I identified over the fact that we're both Star Trek fans, and we started talking very early on. In fact, there was a manuscript in which I was going to dedicate a species to another Star Trek character, who I won't mention right now because of other plans. But in any case, another Star Trek character that we wanted to name, and then we got talking about how nice it would be if we could name a series of frogs after, or a series of organisms after various Star Trek captains. How beautiful it would be for like the consistency of a thing, and obviously it's also very fun for for the media and whatever. And so we've been looking actively for an opportunity to do this for a long time, very long time. And finally here one has presented itself that is not only like suitable in terms of the number of taxa that are being described, but also very suitable in terms of the actual frogs that we've chosen. So these are Bufus, which are tree frogs from Madagascar. It's a Madagascar endemic subfamily, Bufinae, within the Madagascar endemic family, Mantellidae. And there is also one Bufus species found on a nearby island, on the island of Mayotte. There is Bufus nauticus. But in general, all other Bufus species are found on Madagascar. These are tree frogs that have become tree frogs independently from their sister group, the Rhacophoras, which of course also contains several tree frogs. But in any case, Bufus has lots of interesting species, and one of the things that Bufus do particularly nicely or well is that they have very strong bioacoustic differences between the species. And these frogs have particularly different calls than each other. So that's basically the only way you can tell them apart. Morphologically, they are practically indistinguishable. There are some differences in body size, but that's basically it. But they are absolutely beautiful frogs. Let me just show you a picture. I'll hold one up to the camera here for those of you who are watching on YouTube. Here are some beautiful pictures of some Bufus species. And these guys, this, for instance, is Bufus kirki. And you can see they've got these very bright colored eyes. The eyes are kind of orange in the middle and then red around the sides, and then blue in a ring outside of the iris. So they're absolutely stunning eyes. Bufus are often, the common name is bright-eyed frogs. So that's where the name comes from. They just have these very brightly colored
They are all also
eyes.
identical in coloration?
They look exactly the same in coloration. There's very subtle differences. Some of them sometimes have red and some of them never have red. But like it's very, very, very difficult to tell them apart until you start to listen to the calls. And when you listen to the calls, you can tell quite strongly the differences between the species because they emit these piercingly high-pitched whistles that come in a series that is not unlike the communicator beeps or the boatswain whistle that is from Star Trek. So it's these very high... I can actually insert audio file here. Beep, 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 beep. So there you have heard some of these incredible calls of the species that we've described, which are, very different from each other. And I've been involved in discovering several of these species across the different places that they occur. And it's just it's so much fun.
Damn it, Jim, I'm a Bufus, not a...
Yes, exactly. Yeah, so I know that we're probably going to get a little bit of flack for using, pop culture to name some species, but it adds the whimsy that I think is missing from so much of science. And of course, it will also get us some social media traction. And we're hoping you may know, listener, whether or not this has succeeded, but we're also hoping to get a bit of traction from the actors who've played the the captains themselves. So
Nice.
we're in attempts to reach out to various agents, etc., and get some attraction there. And of course, not only is this a bit of a publicity stunt, which of course it very obviously is, but that publicity can help us to gain, I suppose, notoriety, but more importantly, a little bit of funding, for instance, for conservation of these things. And some of them are found. So for instance, in 2015, 2016, I went to these tiny forest fragments that the local people had names for. One of them is called Andranonafindra, and the other one is, so they're anyway, Bevitanyuno is the other one. And they sit right along the road in these teeny tiny little fragments of forest they're nestled into these little valleys that go up the side. And essentially, if those things are still there today, they might not be there in the next 5 years, or 10 years at least. And inside those forests, I found these populations of a previously totally unknown species, that is pikey, I believe, which is an absolute... Oh, no, sorry, it wasn't pikey. Oh, I have to check. Which one was it? Beep, 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 beep. This one. Archery. Yes, Bufus archery, which is another one of these very beautiful little brown frogs. These things are pretty small. They're only about, for the American listeners, about 1 inch long, 2 and a bit centimeters. So very small little tree frogs. And
like our kind of like our spring peepers here.
A lot like your spring peepers, yes. Also in appearance, quite similar to the spring peeper. They can even have a little bit of an X on the back.
Nice.
And they're gorgeous little tiny frogs. And those things are only found in those tiny forest patches, as far as we know. And there's a decent chance that they are, on the cusp of extinction, because that area is being actively mined. And when I was there with these other guys, they were saying, we've been traveling up this road. Last time we were here 5 years ago, this was all covered in forest, and now there's nothing left. So the conservation plight of some of these things is pretty dire. So bringing attention to them by means of giving them some silly names is perhaps warranted, because it can help to motivate these kinds of actions. Yeah. So,
I want to save them all, Mark.
Yeah.
Let's go. I'll meet you there in November, and I'll just stuff my pockets and.
Yeah, I mean, well, we have to talk about conservation breeding at some point on a future episode, because I think that is something that's really interesting. Where there's a German organization, for instance, that is very proactive in that field with some success. There are other. There are ways for people to get involved in things that become managed at that level. So, for instance, I mentioned before the break that we've tried to get some funding to get the genome of the world's smallest tortoise. And that is in collaboration with the people who manage, or the guy who manages the stud book for the species, because they are at the point where they have an active stud book. So one of these management books that helps to get an idea. And it is possible for private breeders of sufficient repute and experience to actually get involved with stud books of that kind.
There's quite a lot in Germany, actually.
Yes.
I know. Yeah.
Yes. Very popular and active in Germany. But yes, for frogs in particular, it can be quite challenging. But pikey might be one of the species that is where that would be kind of at least on the agenda, would be to go and find out is it only there, or is it in nearby forest fragments. There's no reason it wouldn't be in the nearby forest fragments, but we don't know elevationally, where does it stop, where does it start, and how much actual area is there, and are those things... So these things, they only live along very fast-flowing water, and that's probably the reason that they have these extremely high-pitched calls. That they're trying to escape basically the noise of the water, so they go for very high-pitched calls that are quite distinct, even against a very loud background. And but they are characterized by very specialized tadpoles with sucking mouth parts that keep them attached to the rocks in this fast-flowing water. So it's possible, however, nonetheless, that anything upstream, downstream from these areas is getting populated by these frogs too. And it's a question of, do we have this thing of, uphill dispersal then of the adults, where they lay up and get washed down a little bit and then over time try to move up again? Or are they simply completely isolated as soon as they lose those connections, in which case, that would be a species that is really on the brink of extinction. So, yeah, I just wanted to bring that up because it's hot news, which is very exciting.
It is. It is really cool.
Especially as a Trekkie. Anyway,
so I have a question that's related to this, but it's kind of, do we know where the name of the genus comes from, Buphis?
No. So, we know who made it, which was Johann von Tschudi, who I believe was like 21 or 22 when he published what was at the time an absolutely revolutionary paper on the systematics of frogs, and he named Buphis for Buphis goodoti for the very first time. And he seems to have used quite a few words that he simply found euphonic. What I am guessing is that ophis is an attempt to reference the eyes, and that it was perhaps talking about cow eyes, bow as in boss, or something along those lines.
See, because I was thinking,
sure.
like, why? So my brain doesn't go into pronouncing it Buphis. I would go like Bophis.
That's how the Germans pronounce it too, Bo-ophis.
Yeah, I think it would be Bophis, not Buphis, because in my mind it's so similar to Buphis, the toad genus. That it makes no sense to pronounce it that way. I would pronounce it Bophis, like I would say, like because it's written Boophis.
Yes. Yeah.
And the other thing that my paleo artist brain just went, because I'm looking at photos of them, and it's insane the amount of similarities they have to some neotropical hylids in coloration. It's
very much.
People talk about the similarities between like emerald tree boas and like green tree pythons, but this is even more similar.
Yeah, the convergence is crazy.
Yeah. And I was thinking, because you were mentioning about the X, they have sometime on the back, and my paleo artist brain just went, crazy, because that is a pattern that is so common in frogs around the world. You find the same pattern, that it would be great if somebody would, at least try to decipher why that pattern is so common and why it keeps repeating in so many different lineages.
I'm super curious about this. I think there's this really interesting, basically a subset of pre-patterned dermal modules in frogs that are extremely common and are, as I say, pre-patterned. So there's basically a limited set that you can choose from. And if you're going to develop a color patch in one place, then there's a decent chance that it will be on one of these preordained areas. And so that's really, really cool. So there's, for instance, what we have called in our work on stump toad frogs, or stumpfia, we've called it a teddy bear marking, which has then been adopted by various other people, because it has this, it's at the eyes are here, if you're watching the video, but if you're not, you have to imagine you've got two, behind the eyes, you essentially have the ears of the teddy bear, and then you have a kind of a diamond shape, and then goes straight, and then it kind of bends outward, and you have this very characteristic, it's not an hourglass, and that's the reason we call it a teddy bear. And this happens again and again. We see it across many different microhylids, but also beyond the microhylids, we have these kind of teddy bear shaped markings, which is really super weird. Like, why would that be a consistent feature?
By the way, the same happens in lizards and snakes.
Yeah,
I think people have to, that's what I look for when I'm reconstructing stuff, because you do see patterns, and I pay a close attention to that. But I think it would be a great, it would be a great area to study, but I would also think it would be a very difficult thing to study, because I'm sure that you have to find out what genes are, producing pattern in different species.
It needs to be, and it needs to be done at a full developmental scale, comparative developmental work. it's not so super easy, but is of course definitely possible. And now, especially with our means of flagging transcription with various different markers, it's certainly very approachable. In fact, I'd be surprised if someone's not already working on that. But I'm not aware of it at the Anura-wide scale where I think it would be really appropriate. And I think frogs are a particularly well-suited model system because you have access to the embryos in most cases in the form of the tadpoles, and these patterns are probably mostly not laid in tadpole phase, but actually emerge as the tadpole is undergoing the metamorphosis. So some, like tadpoles, sometimes have very amazing color changes as they develop. Zebra-shaped, like zebra-colored tadpoles developing into totally brown frogs and stuff. There's all kinds of weird stuff.
I mean, if you're going to change your morphology completely, you might as well change your coloration.
Yeah, well, yes and no, because like your skin is still your skin. Like I don't know what happens to the skin cells that were skin cells before, if they become further differentiated or if they're simply replaced as it transitions. I don't know how that works. So it's a super interesting, and you know, there's also this phenomenon that in some cases, the vertical line of the frog, which in some groups of frogs, so the mid dorsal stripe, which in some groups of frogs has very simple Mendelian inheritance, is also often apparently correlated with developmental time. So faster developing frogs or the racing stripe frogs tend to develop faster, which is super weird. And like, why would those two things be correlated with one another? That doesn't seem, other than the fact that it's a racing stripe.
The stripes make you go fast.
Yeah, stripes make you go fast. I think that's the only reason. No, but it's.
Stripes are something that happens in everything that tends to go fast.
It's true. It's true. But like developmental biology is not a thing that's like zoo. You know. Anyway. So yeah, no,
sorry for the tangent.
No, totally, but I think it's a interesting, super interesting tangent because I do agree with you. One of the other things that's really interesting with these frogs is asymmetry, which is not so common in terms of color in the biological world. But these frogs quite frequently, sorry for sounds of flipping paper here, will have patches that look like lichen on the back, and those lichen-like patches are asymmetrical. So they've got these very weird, if you're watching the video feed on YouTube, you can see at this point, there's one individual that has these very distinctive blotches on the back of kind of reddish color that are not at all symmetrical with one another.
Maybe that's a Madagascar thing, because you got Europlatus.
Europlatus are a whole other kettle of fish in terms of coloration. I love every time I see someone online. Stuff like that also happens
in neotropical hillocks.
Yeah, I do love it when.
What eats the bowfish species?
Primary predators are probably snakes, but we know that basically anything will eat a good frog. So there definitely have been records of them being eaten by mantis and by scolopenders and birds and other frogs, and
they're not they don't have any toxins, right? They're not
they are probably not toxic. Yeah. And they don't secrete, so they, unlike Mantellines, the sister subfamily, they don't have very distinctive femoral glands, so the femoral glands are the kind of a hallmark of the Mantelline frogs. They have these very distinctive huge macro glands that emit pheromones and various other chemicals, and those are not present in Bufinae. That's kind of one of the things that distinguishes these two groups. So they're certainly not doing as much with smell as the others, and that maybe is related to the fact that they're so very vocal. And some of the frogs, I would also say, are vocal in a way that is very unfrog-like. So these are unfrog-like in the sense that the sounds that they're making sound a lot like a machine or electronic equipment or like a crazy bird or something.
Yeah, I'd argue. Well, that's also similar to spring peepers. I mean, they don't sound frog-like at all either.
That's true. Yeah,
and I've had people mistake them for birds. So,
but spring peeper does tend to release like single peeps, right?
Right. So this is a prolonged.
Yeah. So what these do is they release this this very dynamic chain of sounds where it's like long, short, 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 short. So it is like a very weird sound progression.
And
but
also I think what is frog-like or not depends on what part of the world you're from.
Well,
That's
definitely,
true.
definitely.
That's well. It's not to go on another tangent, but I have red-eyed tree frogs in my room downstairs, and I don't know if you've heard them, but they sound almost like a cackle.
Yeah.
And I didn't tell my wife about them for a while, and she was in the basement where they are and doing laundry, and they started going off, and she was like, "What is that?" And
Yeah. I mean, so what I was going to say about boofers is that there are some species that have truly bird-like calls where they actually are super variable and they just do like weird series of pops and cracks and all kinds of different things. These are not like that. They do seem to emit basically contact calls or small communication calls that are not their huge their high-pitched whistles that are more like a beep every now and then as they're walking around in the treetops. But
is it only
for the
males?
most part, their advertisement call is very stereotyped, which is unlike many of the others. And it's only males.
Yeah.
Yeah. Cool. Well, now that ambulance has passed us by in the background over here, we can move on to talk about another very cool paper that is also... So everything is hot off the press here. So this one has just dropped in Biology Letters by Lindsay Swierk. Long-time listeners may recognize that name. The title is Novel Rebreathing Adaptation Extends Dive Time in a Semi-Aquatic Lizard. And I think, I do not remember which episode it was, many, many, many moons ago, we covered on this podcast the basically original discovery of the fact that there are diving anoles that are actually making a little diving bell for themselves.
Or anoles.
Exactly. Bring back the old fight.
I mean, it's in the dictionary, people. Just look anoles in the dictionary. It's going to tell you anoles.
Yes. Anyway, these lizards have, for those of you who are, for the uninitiated, these are anoles or anoles that, so that's one group of lizards, formerly the family Dactyloidae, now in the family Anoleidae, which simplifies things substantially.
A family composed of one genus.
A family composed entirely of one genus because people can't be bothered to update their own taxonomy. That's a debate that I think we've talked about before.
We have,
but in any case, if someone were to divide them, they would not put this in Anolis anymore.
No, it would be Norops.
It would be Norops aquaticus, yes, quite. And some people treat it in the subgenus Norops as well, which would at least be a first step in the right direction. So, in any case...
So, Norops are known as the beta anoles because they used to be divided in alpha anoles and beta anoles because of their caudal break. You know where the septa have a break? They differentiate in that. So, beta anoles, like Norops, have a preset break in the tail. The alpha anoles don't have that.
I only like the Sigma anoles.
Yeah, for real. I'm more like a West Side Anolis, East Side Anolis type of guy. Anyway, yeah. So
it's the Skibidi Anoles.
So what these silly little lizards do is they dive into the water. They live along the streams, right? And they dive into the water, and then what Lindsay had originally discovered, or Lindsay and team had originally discovered, was that they sit down there and their body seems to be covered in this thin film of air, and then the anole gets or the lizard gets a little bubble over the body that, or especially like pooling over the nostrils, that expands and contracts. And at the time, I remember this was back in the day where we had this thing called Twitter. You may remember it. There was so much excitement and debate and stuff about this because, there was so much excitement and debate about this because it was first of all, so novel and so interesting. And there was a lot of dispute or discussion about, like, how is this working? What is it doing? Is it actually expanding their diving time and everything? And, well, what Lindsay has now, or what Dr. Swierk has now set out to do, is to answer that question. And I think we can summarize the answer as yes. So it's actually a really, really cool study. I don't know if one of you guys want to expound upon the methods that she took, because the simplicity is just beautiful. Because essentially, I guess I'll do it. So what she did was took an emollient, so basically something that will stop water from beating on you, I guess that's right, and applied it to half of the lizards, and the other half were brushed with water, so you had a sham group, and then put them into water, and those, of course, where the emollient had been applied, the water, the bubble did not form, and those that were just the sham treatment, the bubble formed. And then, by simply measuring the time and correcting time and correcting for body mass that the lizards stayed underwater, was able to test for whether or not the bubble formation allowed the lizards to stay underwater longer than when they weren't able to make those bubbles. And the answer is substantially and significantly. So yeah, it's a diving bell, which is crazy.
I mean, in the video, you can even see how the bubble diminishes in size and increases in size as the lizard breathes. I've always got the feeling, I mean, we just found that out. I remember the first time I saw that in a video was a long time ago, which is when this whole thing comes from. It's from a video from a friend of mine that was done for a documentary quite a long time ago. Like, I would say like 5-6 years ago.
Yeah,
6 or 7 years ago.
We had already started the show.
Yeah. We talked about it then. And I always got the feeling that this is probably a lot more common than just in these diving anoles. I think that probably is the same for other groups of lizards who are semi-aquatic, because they all tend to have these rugose scales that probably help them trap these air bubbles. And I think it's easier to observe with anoles because they are smaller, but I think this probably happens more often in other groups of lizards.
Do you think crocodiles would be able to do something like that?
I doubt it.
I think, yeah, probably not.
Maybe something surface tension like related is happening too. So the bigger you go, maybe the less.
Well, the scales of SquaMates and crocodilians are completely different, and the what you see on top of on a crocodile's head is just basically a single scale covering the whole thing that is cracked. So it's not. I think when you see in lizards, you see a lot of times some of the scales face a direction. Like,
yeah.
I would imagine that has something to do with that. Like the way I know that water follows those scales. Like when the rain falls on a mop-headed lizard, like a Eurasintron, I know that that happens the same with some agamids. It basically directs the water to the mouth, no matter where they fall. Yeah,
Moloch does that. Yeah.
So I think that might be happening the same with these air bubbles. It's something definitely worth studying. I think we're just starting to see that of how that works.
I've watched my caiman lizard go in and out of the water a lot, but no, there doesn't seem to be any. It doesn't have the same property as the anole's skin does.
Yeah, because they have smooth scales.
Yeah, well, it's rugo. I mean, it's like,
keel on the back, but on the face, but on the head, they have they have actually plates. They have large plates that are smooth.
Yeah.
I mean, it wouldn't surprise me if there is a body size limit here.
Probably.
You essentially, so we know this, surface area is scales dramatically with body mass, right? Or it actually becomes smaller as you get bigger, so it gets larger as you get smaller, and that's very significant because it basically would indicate that there is a point at which this wouldn't make any sense as you get too big, and then you even if you can cover your entire body in a bubble, which is for all practical purposes essentially what salamanders and frogs do. There's only so big you can get, and also be able to have an effective exchange. And of course, lizards have minimal oxygen exchange over the skin. The dominant proportion of that happens over the lungs, and then it somehow has to have an exchange system to get there. And there's no pump here. So I think there's a really significant thing. I just went back into the archives here. So December 2018, so of course 6 years ago, you were right, Gabriel, is the post on Anole Annals by Lindsay showing this video of the underwater breathing that then cascaded into this whole Twitter discussion, and then there was a subsequent publication related to that in Herpetological Review, I think in March 2019. And then there was another paper that came out in Current Biology in 2021 by Christopher K. Boccia and colleagues, called Repeated Evolution of Underwater Rebreathing in Diving Anolis Lizards, where they showed that this does seem to happen across a bunch of different diving anoles. And it would not at all surprise me if this also happens in diving skinks, for instance, where there are quite a lot of medium-sized.
Same, and I would, for herpetologists listening to this, if you want to go and research other groups that may do this, I would think that you should look at skinks. You should look at gymnophthalmidae that are semi-aquatic, like Echinosaura or Neostichurus or Podamites. And you should look also at tropidurids, like the mop-headed lizards. And I think if you want to check if this is a size-related thing, you should look at basilisks and sailfin lizards to see how that works, because both groups have rugose scales on the head. So it will be interesting to check if that does happen in adults of those species, and also if it happens in juveniles only and not in adults.
Yeah. I would also say water dragons are an obvious agamid to add to the
sailfin.
I would say Tribolonotus.
Yeah, Tribolonotus would be super cool. Because they have that known association with water bodies. But yeah, I mean,
there's a Madagascar skink too, I think.
Yeah, but they're huge. So we don't yet know of any small aquatic skinks in Madagascar. All of them are like chunky boys.
Yeah.
Which is, of course, in itself very, very interesting, and they're doing something else. But so this is obviously a super cool, very simple but beautiful study. That's kind of the modus operandum of biology letters. It's like simplicity. They're short papers. You've done a thing, but the results are significant. And I think here, those results are really significant because you're showing functionality. Whenever you try to publish on something like this, and I can talk about this until the cows come home related to UV fluorescence, they want to have functionality. And if you can show functionality, then you're golden, right? And in this case, basically, this is the experiment that it takes to demonstrate that this has an actual function.
Be right back, guys. I'm going to go put my crown giants underwater.
Yeah. Yes, I think that they might not be thrilled about being cast into the sea.
They're not thrilled about anything, man.
No, that's definitely true. Okay. And then, finally, we have one more paper with which to entertain you, you lot, which is, Gabriel, do you want to do the honors?
Yes. The paper is called Once Upon a Time, Exploring the Biogeographic History of the Largest Endemic Lizard Family in the Neotropics, Squamata Gymnophthalmidae, which is one of my favorite lizard groups. And it's by Ribeiro Júnior, by Juan Vásquez-Restrepo, Marco Ribeiro Júnior, and Santiago Sánchez-Pacheco. It's a very, very cool study, although I have a kind of a little bit of a problem with it. Because
Gabriel, do you ever not have a problem with a paper that we talk about?
For those of you playing SquaMates bingo at home, We've mentioned gymnophthalmids,
Yes.
and Gabriel has a problem with something, so you can drink.
Well,
I
yeah.
also was very close to mentioning the word Sollbruchstelle as the German word for the place that it breaks on the tail, an intended breaking point. And now I've said it, you have to drink as well. But you may have bingo, so congratulations. Anyway, go on. What's your gripe?
So because the paper said that it's in the title that is the largest endemic lizard family in the Neotropics, which is, it is true. They just point out that it is the third largest lizard family in the New World after anoles, of course, Anolidae, which has close to 340 species, Liolamidae, which is this basically South American versions of, for North American listeners, fence lizards is the South American version of that, but there are a lot more.
Okay. So they're like Stegosaurus. Okay.
So like spiny lizards, fence lizards, but they are a lot more varied and a lot more diverse, and they live throughout the Andes and southern South America.
Lots
and those
of
are
debate about their taxonomy.
Yeah, they are. That family has 350 species and about 3 genera. And then Gymnophthalmidae with close to 300 species. The thing is that they say that Gymnophthalmidae is the largest neotropical family, which is not entirely true because the Neotropics does involve all South America, so that will make it the second after Liolamidae. I don't know what they make that
by volume.
Yeah, I don't know what they call the Neotropics.
I was also thinking by mass.
Yeah, right. The Neotropical region involves all Central America, well, all Central America and even southern Florida, and all of South America. So I don't know
Not
what
to mention the Caribbean, which is also, at least in some maps, is included as well.
Yeah, definitely. So I don't know why they are, because I understand anoles, because anoles, just one species, carolinensis, goes out of that region because it is distributed throughout the southeastern United States. But the Liolamids are all within South America, so I don't understand why they make that point. But in any case,
maybe they are Liolamus deniers and don't allow the taxonomic split, or perhaps they are just taking into account how incredibly underestimated the diversity of Gymies is.
But so you can say about Liolamids and...
No, Liolamus have been oversplit to hell. At least if you were to believe some of the authors.
I don't think so. I think they are probably undersplit. But in any case... I would say that the paper otherwise I think is wonderful, and it's great because they have a phylogeny that involves 184 species and 54 genera. That is, that's a very good number.
Half.
Yeah, that's 66% of all the species and 96% of all genera. So it's pretty good phylogeny. It's the best phylogeny I've seen of the group. And they do a great job at talking about the biogeography. And they point out that probably the origin of the family, which dates back to the Cretaceous, to the late Cretaceous, is in the Guyana Shield. And for those of you who don't know what the Guyana Shield is in southern Venezuela and the Guyanas and northern Brazil, there is this upland region based on tabletop mountains called tepuis, which are basically islands. Each of those mountains is an island. And on top of those mountains, basically every single species of plant, animals is endemic to that single mountaintop. So they think that the oldest lineages of Gymnophthalmids come from this region, which is different to what had been proposed before, which it was believed that there was a southern to northern they were thought to start in the southern Andean region and diversified as they went up north. But that's not what this paper shows, and I think it's pretty good, robust finding. And also they found interesting stuff like there was probably not a connection between the Atlantic forest in southern Brazil and the Amazon. but probably the species, the connections were, there was never a connection between those both areas, like a single forest that connected both areas, but probably there was a connection through northern South America that went down to the Atlantic forest, which I thought was very, very interesting. So they divided in this paper the South America and Central America into basically 12 bioregions, which are commonly used. And they think that these bioregions work very well for gymnophthalmids, especially as barriers, because gymnophthalmids tend to be small lizards, which, small animals do respect those ecological barriers more than, that's one of the things that I always complain when people talk about, well, T-Rex was found only in this side. Yeah, but it's a huge animal. It probably had a wide distribution. So, bigger animals tend to have big distributions. Mm-hmm. Small animals tend to have smaller distributions, and they are more usually respect its biological barriers a lot more.
I think it's really interesting that they divide, the Amazonia is split off from Cerrado, which I think to a great extent makes sense, and Atlantic forest. But on the other hand, like if you go and look at their map, those habitats are super interdigitated. So like A, F, and C in their map are like there's whole areas of F that are completely surrounded by C. So there's like the Atlantic forest being surrounding areas of Caatinga, and we know, of course, that Cerrado, and we know that Cerrado habitat is very, very different from Atlantic forest habitat in some respects. But I'm kind of curious as to
many respects.
How is that supposed to be a biogeographic region when they are like so strongly overlapping?
Well, no, I mean, Cerrado is basically grasslands. When you see,
but it's grassland today, and I don't know how
These are pretty old grasslands. They even have some like species that are like found that are like sand specialists and stuff like that. There, that you kind of need a long time to evolve into like those type of. There are microteids, like gymnophthalmids in that area that are like sand specialists and lot of interesting stuff. So the biogeographic regions and those are biogeographic regions that a lot of stuff in South America.
Oh, definitely. No, I know that as well. But I feel like I always have struggled a little bit with things like this because when you do them based on the modern map, and you don't have the individual maps along the way that are paleoclimate inference. And I might be wrong. They might have missed something about having used paleoclimate, but I don't think that they have. Without the paleoclimate and without having a paleo habitat for many of these regions, it's very difficult to inform.
But these are paleoclimate maps from South America are well known from even recent.
Are they integrated here?
I would imagine I'm not sure, but they do talk about it's been hypothesized for a long time that there was a connection. The Amazonian forest at some point extended all the way down to the Atlantic forest. And that does not seem to be the case. And they say that because we do know that there are some relationship. There are even some populations of animals that are found in the Amazon that are found also in the Atlantic forest as an isolated area. But that it does not seem that the area that forms the Pantanal and all that area was ever a forest. Right. And in fact, we do know, if I go back to like the late Pleistocene, we know that the Amazonian forest was smaller than it is now. Like the forested areas were smaller. There were a lot more grassland in South America at that time. So
I mean, I'm not bringing this up as a specific gripe against this study because I think this is a problem that is very widespread in studies that are using the software that they've used, which is BioGeoBears, which is a very good and it's like the standard method that we use to inference ancestral range, to infer ancestral range and things. But it suffers with this problem that that for instance, where grasslands, so grass originated 100 million years ago. Grasslands separate, like really started to establish, like Cerrado started to establish around 65 million years ago. And as soon as you're doing inferences older than 65 million years, you start to be like, okay, well, what was it before? And so this is why I flag it as just like, this is something to be aware of. A limitation of one of these methods is inferring things based on current biogeography presumes that current biogeography and historical biogeography are closely attached to one another, which they might not have been in the context. But in this case, do we know how old are the Gymmies inferred to be? What is their ancestral
late
age?
Cretaceous. Late Cretaceous.
Late Cretaceous. So we've got like a crown age.
Yeah, because there are some other subfamilies within Gymnophthalmidae, like Cercosaurinae or Bacchias. They are thought to have an Eocene origin, but the older lineages seem to date back to the late Cretaceous. And as everybody knows, a lot of old stuff is in the Guyana Shield. That area does, have a lot of old lineages. And
you know what it reminds me? It's the Lost World. It's
that's what the Lost World was based on.
Yeah.
Based on.
That's it just occurred to me. It's like that's what that is.
Yeah, that is precisely what that is. I mean, that's what it was based on. I think that's really so. Gymnophthalmidae here is inferred with a crown age, around 80 million years. So pre-existence of grassland per se, or at least cerrado habitat, but. Of course, the more recent divergence would have happened in the presence of those kinds of patterns being there. So, it's just a limitation of all of these methods. But the outcomes here are super, super interesting for sure.
Yeah, and they, I think, because I always had a problem with that model that said that Gymnophthalmidae have started in the Southern Andes and they expanded north, and that is rarely the case for anything else in South America. So this does show the opposite, that they probably started in the Guyana Shield and expanded from there. And the current centers of diversity are still the Guyana Shield, the Central and Northern Andes, and the Amazon for Gymnophthalmidae. And there's still so much more work to be done with Gymnophthalmidae.
Super interesting.
They are very
I do
diverse.
also wonder in this case if, again, something that always comes up with all these things is that you're only able to sample those things that have survived, right? And there is a bias of things that survive, especially with the KPG event in the middle of this time window, where we could be missing something that would completely contradict any of these things. It's good that it matches like the overall expectation and supports it better than previous work that has been there. But there is always this question of, did everything actually arise in the actually diversify from the Guyana Shield? Like, was it a refuge from which everything arose, or did we simply have biased survival of the things that have arisen elsewhere, and our ability to detect that stuff is really limited.
Yeah, and so far as we know, the older lineages are still in the Guyana Shield. So that's as best as you can answer that question realistically.
So that's one of the things about these methods, like because your oldest lineages are in the Guyana Shield, it will infer that it came from the Guyana Shield. That is not the same thing.
Well, that's
as
not exactly what they say here.
But
that's not exactly what they say here. There's a lot more things. Like, for example, there's the center of diversity, which is still something that is always used to know where a lineage is from, is still the Guyana Shield. I mean, there is a higher number of
that makes a big difference.
Of diverse lineages, not just taxa, but diverse lineages in that region. So I think that's important to that's important to keep in mind.
Yeah. Yeah, I should also like to pay compliments to the authors for their beautiful figures, because they are gorgeous, like very, very clear, nicely illustrated, nicely cut so that they're easily legible. Like really excellent job on something that I know from personal experience is not easy to do. Really, really cool.
So you're saying Edgar Rice Burroughs was right about everything.
Yeah, well, there was some significant, you know, what's his face? The guy who wrote the books.
Edgar Rice Burroughs?
No. What's his name? Who wrote
what book?
The Adventures of Sherlock Holmes?
Oh, Conan Doyle.
Sir Arthur Conan Doyle.
Sir Arthur Conan Doyle. Thank you very much. He was right about everything.
Yes.
Yes.
Spiritualism, all of it. It was all correct.
Thank you for tuning in to this episode. No, but genuinely, I think, you know, it's fantastic the work that these guys have been doing on these lizards. I think one thing to mention here is that the phylogeny behind this whole analysis is actually published separately and beforehand. It's in this paper with the lovely title Alice in Lizardland, assessing the spatiotemporal diversification rates and morphological evolution in the highly diverse microteiid lizards. I think that they need to adopt the title, the word Gymmies. We should really have
far,
t-shirts.
we are the only ones to adopt that, but yes.
But it's coming. It's coming, my friends. We can make some t-shirts or something and create brand identity for the Gymmies. They desperately need it.
They need it. They need it. They're such a cool group that most people don't know anything about.
Yes. So, anyway, I think with that, we can say, I mean, what a tour de force of some very cool stuff that's going on. And I hope that you listeners have enjoyed everything that we have to say. And I suppose, unless anybody has any final things to add, we can wrap up. So, thank you for listening. I don't remember how to wrap up the show. I can only find introduction here. Hey, Ethan, where can one find you on the internet?
At Black Mud Puppy, pretty much everywhere. I'm mostly on Blue Sky these days, but you know,
those guys are bluer.
Yeah, and say hi to me. I've got nothing.
Do it.
I've got literally nothing going on. I'm sure.
Only a whole comic to write and draw and make.
No, just that.
Oh, God. Hiral, where can one find you on the internet?
I am mostly on Instagram these days, so at Hiral's Hive. And then, that'll be great.
Excellent. Gabriel, how about you?
You can find me on X, Twitter, Instagram, Blue Sky, at Serpent Illus, and on my website, gabrielughetto.com. And I also have a Patreon. You can also find me at my Patreon at Serpent Illus if you want to support.
Gabriel, Ethan, you have a Patreon as well, right?
Yeah, it's mainly a pin and sticker club these days, but
well,
yes.
Cool. You can also find Ethan on Patreon. You can follow me at Mark Scherz, MarkScherz, on all of the social medias. Predominantly, I am not on X anymore, although occasionally I do tweet there. I am mostly, I guess, also on Blue Sky, and I sometimes on Instagram, but mostly I am on Tumblr. markscherz.tumblr.com. Come join the Frog Club. It's cool. You can follow the podcast at SquaMates Pod everywhere that you find social media things. Sometimes, very rarely, we post something very exciting. Occasionally, you can find us talking to you through your ear holes. You can find that on youtube.com/@SquaMatesPod or /SquaMatesPod. Not sure. You tell me. On all of the places where you listen to podcasts. If you are listening to this podcast, you have found us. Congratulations, well done. You can find our show notes, which are sometimes extensive and contain all of the references and all the other things at SquaMatesPod squamatespod@gmail.com. You can send us email at squamatespod@gmail.com. We'll be very happy to hear from you. Tell us if you like the show. Tell us if you'd like us to talk about something in particular. We know that there are some requests that we will eventually fulfill. But yes, we are always just happy to hear from the fans. Leave us a comment. Watch along live on YouTube. We love the attention.
Spread the word. Spread the word of the Gymmies.
Spread the word of Gymies, the great
Be a Gymmie
Gymmies.
evangelist.
Be a Gymmie evangelist. And with that, my friends, we'll leave you as we always do by saying, Hakuna
Squamata.